Now we are going to talk about the significance of the HMP shunt. So before starting this topic, significance of HMP shunt, I want you to study the various steps of HMP shunt which I have discussed it in previous video. So after knowing the various reaction, oxidative and non-oxidative reaction of the HMP shunt, if you have noted down that reaction, then only proceed to this video to see the significance of the HMP shunt. So as I have already told, the HMP shunt generates two important products. One is pentoses, the ribose, and NADPH. There is the generation of the ribose 5-phosphate, and I want to start with the importance of the pentoses, which is ribose 5-phosphate. The ribose 5-phosphate is used in the synthesis of nucleic acid and nucleotide. This is the one of the very important significance of the HMP shunt. The product of the HMP shunt, which is ribose 5-phosphate, is used in the synthesis of the nucleic acid and nucleotide. Another important product is NADPH. There are various number of number of functions of the NADPH. The NADPH is required for the reductive biosynthesis of the fatty acid and the steroid. This is the one of the very important function of the NADPH. The NADPH is also required for the synthesis of certain amino acid involving the enzyme glutamate dehydrogenase. And if you know that in the metabolism in the body, in the cell, there is continuous production of the hydrogen peroxide in the living cell, and the hydrogen peroxide can chemically damage the unsaturated lipids, proteins, and DNA, and this is prevented by the antioxidant reaction involving the NADPH. The antioxidant reaction involving the glutathione peroxidase and the glutathione reductase. So this NADPH acts as the antioxidant function, helps in the antioxidants reaction, which is used in the neutralizing the hydrogen peroxide, so that the chemical damage would be reduced. This I will discuss detail in another video in the glucose 6-phosphate dehydrogenase deficiency. Here you only have to know that the product of the HMP shunt, the NADPH, takes part in preventing the chemical damage of the hydrogen peroxide in the living cells. And another importance is microsomal cytochrome P450 that is found in the liver brings about detoxification of the drugs and foreign compounds by the hydroxylation reaction, and that hydroxylation reaction requires NADPH. And phagocytosis in the cell also requires NADPH. So NADPH is one of the important important product. In the RBC, what happens? NADPH maintains the reduced glutathione, and this reduced glutathione is required to preserve the integrity of the RBC membrane. So if there is deficiency of NADPH, then there will be the impaired reduced glutathione, and if there is no reduced glutathione, then the integrity of the cell membrane, the integrity of the membrane of the RBC is disrupted, and the RBC would be deformed if there is less production of the NADPH, or there is any default in the HMP shunt. And increase NADPH, the more number of NADPH is required for the transparency lens. So if there is any problem in generating the NADPH, then the lenses would be opaque. So these are the various importance of the HMP shunt. So HMP shunt mainly generates two products, two main products. One is pentoses and NADPH. The pentoses ribose participate in the synthesis of nucleic acid and nucleotides. The NADPH, however, has various function. Biosynthesis of the fatty acid, steroid, and synthesis of the certain amino acid involving the enzyme glutamate dehydrogenase, The NADPH also helps in the antioxidants reaction to reduce the chemical damage of the hydrogen peroxide, and it also helps in the hydroxylation reaction. The NADPH helps in the phagocytosis, whereas the NADPH helps to maintain the reduced glutathione in the RBG so that the structural integrity of the RBC would be maintained. So NADPH is also found in the lenses. The increased amount of the NADPH helps to maintain the transparency of the lens. So these are the various significance of the HMP shunt that you should know. So in the another video we will talk about the G6PD deficiency. I hope you understood this lecture. Thank you.